Some cells can move from one place to another within the cell's cytoplasm. Cell structures and substances needed to maintain internal stability of the cell show movement. Cells have a cytoskeleton made up of a network of microfibers that direct these movements and give support and shape to cells. Some cell parts are held in place by these fibers. Cytoskeletal fibers are very small, ranging from 6 nanometers to 25 nanometers in diameter. Small in that 1000 nanometers equals 1 micrometer, and 1000 micrometers equals 1 millimeter. Observing Elodea cells with a light microscope shows a type of movement in the cell cytoplasm called cytoplasmic streaming. Cytoplasmic streaming, or cyclosis, is the movement of the liquid in the cytoplasm of these cells. Structures called chloroplasts, the green bodies, are circulated in the cell by this process. This occurs inside the cell membrane outside the vacuole, which is relatively large in plant cells compared to animal cells. These organelles more or less are towed along cytoskeletal fibers by what is called motor proteins. This movement helps move other organelles, nutrients, etc. to all parts of the cell. Slower cytoplasmic streaming can be observed in some protozoans, such as the paramecium. Green food vacuoles can be seen transported throughout the cytoplasm, during which food is digested and its nutrients are distributed throughout the cytoplasm. These food particles are swept into the oral groove of the paramecium by tiny hair-like structures called cilia. The cilia also cover the entire body of the paramecium and enable these cells to move from one place to another. Cilia are made of components of the cytoskeleton that run the entire length of the cilium. The rhythmic motion of the cilia allow these movements. Stentor is also a ciliate in that it has rows of cilia that cover the entire body. They also have different kinds of cilia around the mouth. These cilia are fused into what are called cirri. The cirri make up rows of spiraling membranelles that create currents that move food particles into the cell. Flagellates move by flagella, which are structurally similar to cilia. Movement of both results from the interaction of the same kind of cytoskeletal components. Flagella, however, are longer but fewer thread-like structures, whereas cilia are shorter but more numerous hair-like structures. Flagellates usually have 2 flagella, but normally only one can be seen. Some move by metaboly, where they simply change shape. A euglenoid showing 2 flagella. The amoeba move from one place to another by forming temporary extensions of the cytoplasm called pseudopodia, or false feet. This movement is called amoeboid movement. Pseudopodia are formed by components of the cytoskeleton. These cells are pulled forward by contractile forces of these components. The cytoplasm flows towards the leading edge of the pseudopod. Cell organelles and granules move in the cytoplasm. You can see the granular appearance of the nucleus and the clear, spherical contractile vacuole. The enlarged tail region of the amoeba is called the uroid. The amoeba uses its pseudopodia to capture prey for food. The prey is surrounded by the pseudopodia, which forms a food vacuole. The vacuole circulates in the cytoplasm, where the food is digested by digestive enzymes. Pseudopods of a shelled amoeba. During animal cell mitosis, the centrosomes can be seen, one at each end of the cell. The centrosomes produce the cytoskeletal structure, the spindle. Some of the spindle fibers attach to each chromatid and separate them into new chromosomes. A complete set of chromosomes is pulled to opposite ends of the cell by these fibers. Cytokinesis is the division of the cytoplasm at the end of mitosis. The cell membrane begins to constrict, forming a cleavage furrow controlled by cytoskeletal elements, which eventually pinches the cell in half, separating the two new cells. Plant cells do not have centrosomes, but do produce a spindle that pulls the new chromosomes apart. During cytokinesis in plant cells, a cell plate forms to separate the new cells. Vorticella is a ciliated protozoan, usually attached to the substrate by a contractile stalk. The action of the stalk differs from most other types of movement. The contractile element, a cytoskeletal fibrous structure within the stalk, when stimulated, shortens, resulting in the stalk coiling like a spring. This is the most rapid movement known. Other movements of some protozoa are usually attributed to the action of these cytoskeletal contractile fibril elements. For example, the darting extensions and contractions of the neck of Lacrimaria and the flexible, mobile proboscis of Dileptus. Both body structures are used to come into contact with their prey. Suctorians are protozoa that use specialized tentacles for feeding. The tentacles contain a bundle of cytoskeletal tubules that form a cylinder-shaped canal from the tip of the tentacle to the main body of the suctorian cell. Cytoplasmic contents of its prey pass down the tentacle canal within a long cylindrical food vacuole.